So our final speaker of the morning session is Franziska Michor. I do not know her children, but I do know her work, and we're very lucky to have her here. She's a director of the Dana-Farber Cancer Institute Physical Sciences and Oncology Center in the Center for Cancer Evolution, and she's also a professor at the Harvard School of Public Health. Franziska.
Thank you so much. I feel a little bit as a disadvantage because not only do I not know Matthew's kids, I'm also between you and lunch. So I'm going to try to make it as interesting as possible to keep you in here. So thank you to the organizers for the invitation. So what we do is we look at cancer as a problem in evolutionary biology. This is partly because my background is in evolutionary biology, but partly because I think this is a good way of thinking about cancer, because as Gadi outlined, over the course of the life history of a tumor, multiple alterations, they can be genetic or epigenetic or any other type, are accumulated that then lead to things like drug resistance, evasion of the immune system, and infiltration of other organs. So given that this is a natural selection problem, what we thought we could do is use the tools of evolutionary biology, particularly theoretical evolutionary biology, to describe this problem as a mathematical framework, and then use the tools of math to try to make a difference and try to understand things better. So what we want to do is quantitatively describe how these heterogeneous cell populations evolve over time and accumulate more and more aggressive changes and populate other places, and then use that description to figure out how to, for instance, treat patients better. So what I want to show you today is how we can use this kind of methodology to try to design better treatment administration strategies for cancer patients. So I want to show you two examples. One is for EGFR mutant non-small cell lung cancer, treated with EGFR tyrosine kinase inhibitors, such as erlotinib, and then the next generation inhibitors. And the other one is glioblastoma, treated with brain cancer in combination with radiation in combination with chemotherapy. So this idea came around when I visited a colleague at Memorial Sloan Kettering who was a thoracic surgeon and oncologist, and he visited his patients and he said, "Oh, you have a headache, we should halve the dose," which to a computational analysis person sounds a little bit weird, because if you halve the dose, that could really have tremendous consequences on the evolutionary dynamics of this tumor cell population. So we set out to try to come up with a method that would enable us to understand what the consequences would be for the evolutionary dynamics of the tumor if you just halve the dose. And so Tarceva, or erlotinib, you might know this, is actually administered at 150 milligram orally across the board, unless you have a headache and the dose is halved. So the problem with this is, of course, the way a clinically administered strategy is developed is usually through a 3+3 dose escalation scheme, where you start with a dose that you think is safe based on preclinical data, and then you escalate up until you hit a level that is not tolerated, that leads to dose-limiting toxicity in these patients. And of course, that might be a good idea for some situations, but it certainly doesn't rationally ask or search through the entire space of all possible dose administration schedules what would be the best way of, for instance, minimizing the risk of metastases, or minimizing the chance of resistance, or minimizing the rate of evolution, no matter what the evolutionary outcome would be. And so that could have strong effects. So let's have a little thought experiment. It's very simple. It doesn't even have pharmacokinetics. Let's consider we can administer a drug at two alternative dose administration schedules. One is this low-dose continuous strategy, such as Tarceva, where you give 150 milligram every day, and over time that will kind of reach a steady state and be maintained at this steady state concentration in the bloodstream. What happens in this scenario, if you don't have any resistant cells present, in the simplest possible scenario, is that the number of sensitive cells will be driven to extinction exponentially at a slow rate. That's reflected by this low-dose continuous strategy. If you do something else, like a high-dose pulsed strategy, you actually, by definition, can't maintain this high dose at a constant rate because it's above the maximally tolerated dose at a continuous strategy. So what you have to do is administer a drug holiday, during which the cancer cell population is actually going to expand again because there's less drug present, maybe even no drug present in the system. So then the question is, so what's actually better to do, this one here or this one, or one of the other strategies, or one of the millions of other strategies that we could potentially give to these patients. And how does that matter and depend on the heterogeneity of the tumor cell infiltration with immune system cells, on the pharmacokinetic parameters, on the patient-to-patient variability, on maybe how much they eat or how much they weigh, and so forth. So what we came up with was a very simple first step model. This is based on a stochastic evolutionary process that's called a branching process. It's a stochastic process where during each elementary step, a cell is chosen proportional to fitness to divide. So you can calculate the probability of a cell or a clone increasing in frequency or decreasing in frequency. That depends on the growth and death rates. That can be functions of time, because the drug concentration in the body can be a function of time. And in the simplest case, there's one resistance mechanism that arises with a certain mutation rate. That doesn't have to be a point mutation, could be any kind of alteration, but arises at this rate from the sensitive cell population and also grows or dies exponentially. And what we can then do is parameterize this mathematical model with in vitro data. In this particular example, EGFR mutant non-small cell lung cancer cell lines that are treated in vitro with different concentrations of alatinib, and then we measure growth and death rates of these two cell populations, the sensitive and the resistant cell population, during treatment with these concentrations. Then we couple this cell-based evolutionary model with a system-level pharmacokinetics model. So it becomes a multi-scale description where we have the cell populations proliferating and dying according to those rates that we've specified. But we also have the drug concentration varying in the plasma and also in the tumor, because the dose administration strategy might be different. And then, based on the toxicity constraints that we estimate from clinical data, we basically solve the optimization problem. And that is, in all of those millions of strategies that we could possibly administer, given those that are clinically administrable, what's the one that's best in order to minimize the number of resistant cells at a certain time point? And of course, this is not something that you can do without a mathematical framework, because we can't run any of those 100 millions or however many that would be, those administration schedules. So this is a way to narrow the search space with a computational approach, so that we then validate preclinically and then eventually clinically only maybe a couple of the strategies and not all of them. So this is exactly what we did. We twisted somebody's arm at Memorial Sloan Kettering. To this day, I don't know how we got him to do this. But we started a phase 1 study of twice weekly pulse dose alatinib together with daily low-dose continuous alatinib. So this is what came out of this optimization to be the best thing to do. The pulse, 2 days, 20 micromolar, 5 days, 1 micromolar, even though the 20 micromolar was just a guess that would be clinically administrable, given that the low-dose plus high-dose pulse strategy had never actually been tried in combination. And so it was a single center phase 1 dose escalation study to determine the maximum tolerated dose of this combination and also eventually assess efficacy. And everybody with metastatic EGFR mutant non-small cell lung cancer was allowed. No prior treatment of EGFR tyrosine kinase inhibitors was allowed. We had 34 patients total, 24 in the dose escalation cohort and 10 in the expansion cohort. We determined the MTD to be 1,200 for the pulse, those days 1 to 2, and then 50 milligram apart, or in contrast to the 150 milligram, which is the FDA-approved strategy on the remainder of the days. The waterfall plot was well tolerated. We had 30 subjects evaluable for response. 4 came off because of toxicity and non-compliance. We had overall response rate 74%, median progression-free survival 9.9 months. And very interestingly, very highly variable PK, which is not something we were expecting. So here are the individual PK traces of all the patients that were treated on this trial. And you can see the different dose cohorts based on the high-dose pulse. The low-dose pulse was the same at 50 milligram for every patient for the rest of the week. And you can see that there's high variability. For instance, if you look at these 2 red dots here, those are the 2 patients that received 750 milligram. And you can see that they're almost at the top and almost at the bottom of this. So there is very high interpatient variability in the PK dynamics over time. Which unfortunately led to an underpowering of the study, so that we didn't have enough patients to see a significantly different time until progression of disease due to T790M, which is the main resistance mechanism that we were modeling here. So we're now expanding this cohort to include more patients and that dose administration schedule. What was significant, though, was that we completely prevented progression of disease in patients with brain mets. So this is up to 33% in historical control with the 150 milligram a day dosing strategy. But in this trial, it went to 0. And so we are also expanding with a second set of patients with brain mets to see whether this is actually something that can be replicated and even seen in a larger number of patients. We're now going on to combination treatment. We're looking at next generation EGFR tyrosine kinase inhibitors, such as osimertinib, and also MEK inhibitors, such as selumetinib. There's a particular trial which is called the TETAN trial, which is run by AstraZeneca, also at the Dana-Farber, where they're looking at different combinations between osimertinib and selumetinib, where if we now expand our mathematical modeling framework to look at different resistance mechanisms, one against the EGFR tyrosine kinase inhibitor and one against the MEK inhibitor, to see what the optimum schedule would be. And here what we find with this modeling, which is done by Sean in my lab, is that you can make very big improvements up to 40% above what's being used currently in the TETAN trial if you just mess with the dose administration schedule of osimertinib and selumetinib a little bit. So now we're in discussions with both the PIs at Dana-Farber and also AstraZeneca to try to see whether we can have a third arm where we could validate some of these ideas. We're also extending these methodologies to come up with better parameter estimation using single-cell lineage tracing data. So the problem with some of these in vitro data sets that we have is that we take the population average growth and death rates. But of course, the better way of doing this would be to do time-lapse microscopy of individual cell lineages treated with for instance, osimertinib, where we could then come up with computational analysis techniques that now involves Wall Street math to estimate what the actual distribution of growth and death rates would be estimated from single-cell data. And this is still ongoing. Now, some of the resistance mechanisms that drive response or resistance in individual cancer types are not actually cell intrinsic, such as T790M, which is just a second site mutation in the EGFR tyrosine kinase domain. Some of them are microenvironmentally driven. And so because we were interested in trying to understand and develop some of these computational modeling frameworks that can take into account some of these microenvironmental determinants of resistance, we started investigating glioblastoma and its radiation response. Because it seems as if the radiation response in primary glioblastoma cells really depends on the localization of the cell within the tumor microenvironment, and also which cell type it is. So it turns out that tumor stem-like cells that have high CD133 or nestin expression are localized right next to the blood vessels, right next to the endothelial cells in the perivascular niche of these tumors, and that their radiation response is really modulated by signaling that originates from endothelial cells and makes these tumor cells more protected so that they're largely radio-resistant, versus tumor differentiated cells that are localized further away from the blood vessels are largely radiosensitive. So what we did in this case is we used a mouse model of glioblastoma, particularly of the proneural subtype, a PDGF-driven mouse model, to understand this intratumor heterogeneity and the dynamics of different cell types as they respond to radiation treatment and where they're localized within this perivascular niche. Then we came up with a computational framework that described both the stem cell population and the tumor bulk as they respond to radiation, and characterized how many or what proportion of these cell types actually die in response to radiation. And use that then to again search through the space of all possible dose administration schedules of 10 gray, just for a mouse trial, 10 gray over 1 week, with the constraints of the radiation oncology clinic, which means not before 8 a.m. and not after 5 p.m. and definitely not on the weekend. And we found this optimum here, where we give 1 gray each, Monday at 8, 2, 5, Tuesday at 5, and so forth. And we then validated this in a mouse trial, and what we found here is that there's no significant difference between giving 10 gray either all at once or in standard of care, which is 2 gray every day for 5 days, or what we call a scramble control, which looks similarly ridiculous as this thing here, but actually is predicted not to do any better. But what we found is that this schedule here really leads to significant survival benefits, so much so that it's now non-significantly worse than this pink line here, which is standard, like the red one, but for 2 weeks, for a total of 20 gray. which suggests that if you just mess with the timing at which you administer radiation, at least in this mouse model, you can really make a difference in terms of survival of the mouse. Now, what the mechanism of this seems to be that we actually enrich for this slower proliferating radiation-resistant stem cells, which doesn't sound like such a great thing to do, but at the end, it still increases survival. So maybe this actually is a mechanism of actually increasing survival, even though it might not actually be curative, but neither is any other radiation administration schedule. And this is achieved by timing the radiation fractions in this specific way to maximize the dedifferentiation process and hit them again before they have actually dedifferentiated in the entire way so that they can hide out in this radio-resistant stem cell niche. And this is actually also something that's observed in humans. So if you look at a cohort of patients that received 60 gray radiation plus adjuvant temozolomide, and you measure the CD133 fraction, which is basically a measure for the tumor stem cell population, then you can see that those that have a larger stem cell fraction have a significantly improved survival than those that have a lower stem cell fraction. And so now we're working on clinical implementation of this finding. We are just submitting the IRB protocol for a pilot trial that will have 10 patients, just to see whether it's feasible to get them in at these crazy times and many times a day sometimes. The dose constraints here are to come up with a biologically effective dose such that late responding normal tissues don't have more toxicity than the control arm. And we need to have at least 0.75 gray per dose. And we can have at maximum 3 fractions per day. And we want to reduce the number of fractions as far as possible so that we don't actually have a patient sit in the radiation oncology clinic 24 hours a day. And so what comes out here is that we have identified an optimal schedule for the clinical implementation for 35 gray total. First, we give 28 gray in 7 fractions, 1 fraction a day, and then 9 gray in 3 fractions, so 3 fractions a day at the end. And this maximizes differentiated cell killing per fraction using this hyperfractionation at the start of treatment, and then enriches for the stem cells with this ultrafractionation at the end of treatment to hopefully maximize survival. And in fact, this is not actually the global optimum. If you search through the space of all different 35 gray administration schedules, there's one thing that's a little bit better, and that's this one here. But the problem is it has many more ultra-fractionated days, and it's not really very significantly better. So if you look at fractional volume change over time since the start of treatment, then you see that it's a little bit better, but it's not tremendously better. So we're going with the green one because it's easier to implement. And hopefully we will actually see that this is feasible and then expand this to hopefully a multi-institutional trial where we can prove efficacy of such an alternative dose administration schedule. Which brings me to the last thing I want to show you, and that is, so what happens for upfront treatment? In the recurrent setting where we give the 35 gray, we can get away with just doing radiation. But upfront, we have to combine it with temozolomide because that's standard of care. Now, temozolomide, unlike radiation, is not a field effect, right? It doesn't really matter where exactly the cell sits to determine how much radiation it actually will receive. It only matters whether it's a stem cell or a differentiated cell, and that determines radiosensitivity. Now, with temozolomide, it's different because temozolomide diffuses away from the blood vessel in the middle of the tumor, and then the cells that are closer to the blood vessel necessarily get a larger concentration of temozolomide than something that's further away. So we then came up with a spatially explicit computational model where we have the blood vessel in the middle, so those red cells are the endothelial cells. And we have the stem-like tumor cells around, and at the periphery we have the differentiated tumor cells. And this is again parameterized using this RCAS-TVA model of somatic gene transfer. And now the question is, what's the best way to administer radiation treatment if you also give temozolomide, for instance at 3 p.m. the day before? And if you optimize that, then you come up with something that looks like this. So we're now giving 1 gray Tuesday, 9 and 4, and then so forth. We also predicted, again, a suboptimal to see whether we have predictive power with this computational approach. And all of those get temozolomide 50 mg per kg at 3 p.m. the day before. And when we validated that, again, in the RCAS-TVA model, we found, again, a very and this is just hot off the press, that came out last week. So again, the mice treated with the optimum have a significantly prolonged survival duration than those treated with the suboptimum. And interestingly, the optimum here is not the same as the optimum without temozolomide. So we really have to take into account what other treatment they're receiving and potentially also which subtype of glioblastoma they are, because this is all validated in PDGFB-driven, so the proneural subtype of glioblastoma. So now we're working on applying such methodology not only to the other glioblastoma subtypes, but also other tumor types. We're working on leukemias, we're working on breast cancer, melanoma, all kinds of different things. We are also extending this to combination treatment, like I showed you for the lung cancer case, and we're also very interested in immunotherapy, which of course is an even more complicated question because you need dynamic data of individual cell types as they respond to different things over time. And so we're still working on that. Big shout out to my lab, they're awesome. And then we are just launching this new thing, which is called the Center for Cancer Evolution at the Farber. And if you want to get involved, please do. Thank you.
Great, thank you very much, Franziska. That was fantastic. So let's thank all of our speakers one more time.